So the cell membrane is sometimes called the cytoplasmic membrane or the phospholipid bilayer. It's made out of phospholipids. So in this diagram of phospholipids, you're not seeing the actual chemical composition of them, but they have these gray areas. These are the fatty acid tails. These are hydrophobic, meaning they don't mix with water. Sort of like they're an oil, they're a lipid, they're an oil, they don't mix with water. So this is called the fatty acid tail. And each phospholipid has 2 of these tails. And then this yellow ball is supposed to represent the phosphate head. And these are polar or negatively charged often. So these are hydrophilic. So these are attracted to water. Okay, so we say that phospholipids, so each individual phospholipid, so you have this yellow ball, the phosphate head, that's hydrophilic, and these 2 hydrophobic tails, so that's one phospholipid. We say that they're amphipathic molecules. An amphipathic molecule is a molecule that has a hydrophilic region that's attracted to water and a hydrophobic region that's not attracted to water on the same molecule. So these orient themselves this way because the hydrophilic phosphate heads will face towards the water on the inside of the cell and towards the water on the outside of the cell because it's attracted to water, while the oily hydrophobic tails will face inward towards each other, separating out from water. Okay. So when you have a whole bunch of phospholipids, they tend to arrange themselves into what we call this bilayer. So you can see this bilayer structure where there's a layer of phospholipids here and a layer of phospholipids down here, and the tails face each other, the hydrophobic tails face each other. So I want to emphasize that it's not that lipids repel water. It's really actually that water molecules are attracted to each other so strongly that they exclude lipids, such as oils, by pushing them away so they don't interfere with the relationships the water molecules are having with each other. So let's look at what I mean here. So here in this picture here, it's just a picture of oil and water mixed together. So if you have a mason jar and you fill it half full of oil and half full of water and you mix it up and then you let it settle, the oil and water all separate out from each other. So that's because water is attracted to itself to the exclusion of oil. And to understand this, you have to remember back to the chemistry of water from prereqs. So water molecules are polar. And that means one end, the end with hydrogen, is positively charged, and the other end, the end with oxygen, is negatively charged. So here you have water is H2O, so you have 2 hydrogens and 1 oxygen. This is because oxygen is an electron hog. It doesn't share electrons well with hydrogen. So you might remember that covalent bonds, so a molecule is made of covalent bonds or shared electrons. So the hydrogen and oxygens are held together in one water molecule because they're sharing electrons with each other, right? So there's these electron pairs that are circling both the hydrogen and the oxygen, and the fact that these electrons are circling both of these atoms holds them together. That's a covalent bond. But not all elements share electrons equally because some elements are are stronger attractants of electrons than others. So oxygen is an electron hog. It's a strong attractor of electrons. So when oxygen and hydrogen are sharing electrons with each other, they're not shared equally. These electrons are spending more time around this oxygen than around the hydrogen. So it is shared, but a disproportionate amount of time is spent around the oxygen. Now electrons are negatively charged, so that means that these negatively charged electrons are swirling around this oxygen more often. That's going to give that oxygen a negative charge, but it's sort of flickering because then the electrons go back to the hydrogen again. So this little Greek delta here, you can see this like negative symbol, but it's next to this what's called a little Greek delta. That symbolizes that it's a partial negative charge. It's not a full negative charge. For it to be a full negative charge, the oxygen would have to steal the electrons from hydrogen completely, and they wouldn't be sharing anymore. This is sort of like a flickering or a partial negative charge because the electrons are still being shared, but they go back to the hydrogen sometimes, even though it's not very often, even though they spend a disproportionate amount of time around oxygen. All right, so So you know that each individual water molecule is polar, meaning the two ends are different. There's a negatively charged oxygen and a positively charged hydrogen. Now, opposites attract. So the positive charge of the hydrogen on one water molecule is going to attract the negatively charged oxygen on a different water molecule. So these little dotted lines, they represent those attractions. There's no electrons being shared between this water molecule and that water molecule, but there's an attraction of opposite charges. And that's just like, if you have two magnets and each magnet has a north pole and a south pole, the north pole of one magnet is attracted to the south pole of a different magnet, right? It's not that they're actually bound together, like it's not like they're sharing matter. They're just attracted to each other because of these oppositely charged forces. So it's just the same here. These are attracted to each other like weak magnets. Now, because these are partial charges, the attractions are quite weak. They're easy to break, and that's why water is a liquid at room temperature. So the energy so room temperature has a certain amount of energy in it, heat energy. That small amount of energy is enough to break these bonds. So these water molecules can still flow back and forth past each other. That's why it's a liquid. But if you were to remove all of the energy in the room and make it very cold, that's why water solidifies to ice. And it forms these crystalline structures based on the geometry of these attractions. So you know that water balls up in these little droplets, right? So if you were to wax your skis or wax your car and then it rained, you would see the water bead up and form this little ball. And that's because of this, it's called cohesion. So it's the attractive forces between each of these molecules. They're attracted to each other, so they don't just flatten out. They actually hold some shape, even at room temperature. And so I want to show this video here, and it shows a water droplet in slow motion. So what you'll see is, in this video, a water droplet is going to drop onto sand, and when the water, so you'll watch that happen in slow motion, and when the water droplet hits the sand, the force of impact is going to cause all the water molecules to spread out. So you'll see, the droplets start to smash apart and extend outwards like a flower, but then it all gets sucked. All those water molecules get sucked back in together again. So the droplet starts to spread out because of the force of impact, and then they get all sucked back in and they settle as a droplet. And you can search YouTube for other slow motion videos of water droplets. For example, if you watch slow motion of raindrops hitting a lake, the raindrop will actually bounce on the surface of the water and hold its form for a little while before it then blends in with the lake. So oil and other lipids have no charge. So water molecules aren't attracted to lipids. They're neutral. They have no positive, no negative charge. This means that water molecules will pull each other away from the oil, since the oil has no electric charge to hold the water molecules. So here you can see, because it's opposites attract, right? Like repels, opposites attract, but so that refers to positive and negative forces. Neutral forces aren't related to any of those attractions or repulsions. Okay, so here you can see a lone water molecule lost in a layer of oil. So here, here you can see the two positively charged hydrogens in red and the negatively charged oxygen in blue. So that's H2O. And then, so this is a lone lost water molecule in all this oil. So here's the neutral oil, and then here's the rest of the water down here. You can see this key on the left. So oil at the top, water at the bottom. So this lone water molecule, it's not that oil and the water are repelling each other. It's just that these attractive forces, the opposites attract. So the negatively charged oxygen up here is attracted to these positively charged hydrogens down here. And those attractive forces end up pulling that lone water molecule out of the oil, separating it out so that all the water molecules can stick together. That's the cohesive forces. There's cohesion between water molecules because of the attractions between the positive and negative charges on these polar molecules. So that's really what I want to emphasize. There's nothing that oil and water, yes, they separate. It's not because they're repelling each other. It's that all the water molecules are attracted to each other, and they end up excluding the oil because of that. Right, so there is water inside and outside the cell membrane. The charged, those charged phosphate heads on each phospholipid are attracted to the water on the outside of the cell and on the inside of the cell. And then those oily fatty acid tails face each other away from the water, creating a barrier between the inside and the outside of the cell. Bilayers like this, like the ones that form cell membranes, actually form spontaneously when you mix amphipathic molecules like phospholipids and water. These molecules will spontaneously orient themselves in this way just because of their chemistry. It has, there doesn't need to be a living thing doing this. So if you just take a bunch of phospholipids and you shake them up in water and then you plate them on a slide and then you look at them, watch them under a microscope, you can find YouTube videos of these bilayers forming. It doesn't mean that a cell is spontaneously arising, right? Because there's nothing inside those bilayers, but they will just because of their chemistry, this is what happens when phospholipids and water is mixed, because the phosphate heads are attracted to water, and so they are going to orient themselves to face water no matter what. And then these hydrophobic tails, they're excluded from that relationship, from those attractions between the phosphate heads and the water. And that's really essential because the cell exists in water. Your bodies are mostly water. A cell is mostly built of water. So you want to have a membrane that does not dissolve. It needs to retain some integrity, but you also want a cell membrane that's fluid, the way that, oil is fluid, because that fluidity gives the membrane this dynamic it's just the cell membrane is dynamic. It can move, things can move through it. That allows the cell to move, that allows things to happen in the cell surface. So you want it to be fluid and yet not dissolve in water. So what better way to do that than to make it out of a lipid, like an oil? So the fatty acid tails, that's the oil part, but it's not just a blob of oil, right? It has this interesting structure that allows there to be an inside and an outside, and that's because of those phosphate heads, which organize the oily part so that it's not just a blob. And this chemistry is also responsible for the cell membrane's role as being a selectively permeable barrier. What I mean by selective permeability, the cell membrane only lets some molecules in, but not all molecules. So permeable means that, things can cross it. But selectively permeable means that some things can cross, but not others. So the cell needs to create an internal environment that can retain some consistency, even though the external environment might be changing all over the place. The cell needs to, inside of the cell membrane, the cell has to be able to maintain certain conditions that are compatible with life, like a certain pH, a certain salinity or osmotic potential, a certain amount of nutrients for energy. So those kinds of things are things that the cell needs to regulate. And to do that, it needs to create a barrier between the inside and the outside, because the external environment, the cell can't control that, right? The external environment is just going to be changing all the time, but the internal environment has to remain constant, and that can only happen if there's this barrier. But things need to be able to get across this barrier because the cell needs to bring in nutrients, calories, and it needs to get rid of waste products. And so there needs to be ways for things to cross. So Part of the reason the cell membrane is semi-permeable is because of its chemistry. So the bulk, I know these fatty acid tails and all these diagrams look really short, but the reality is that they are actually quite long relative to the size of these phosphate heads. And so the bulk of the cell membrane is hydrophobic. And so small hydrophobic molecules can just cross directly across the cell membrane. So that's going to be things like atmospheric oxygen is a nonpolar molecule. By nonpolar, I mean it does not have an electromagnetic charge. It's not negative, not positive. It's just neutral. So a nonpolar molecule would be things like carbon dioxide, oxygen. Those are things that can just cross directly through these phospholipids. But another way that it can be selectively permeable is the cell is capable of creating channels that are very selective and that will only allow certain molecules to cross. So for example, if the cell wants to bring in sugars, it can have a channel that will only allow sugars to cross and also only in one direction so that they can only come in, but then they can't escape. Okay, so the sugars are polar. They have an electromagnetic charge. That means they can't, they're not going to cross this phospholipid bilayer. You can just remember, hydrophilic things dissolve in water and they mix with water. Hydrophobic things don't mix with water, but they mix with hydrophobic oils. So hydrophobic mixes with hydrophobic, hydrophilic mixes with hydrophilic. So sugar dissolves in water. It can't cross the hydrophobic barrier here, the oily barrier. So it needs to have a channel to cross, and the cell can decide what kinds of these molecules it's going to make a channel to allow through. So this brings us to how the cell moves things back and forth across the cell membrane, which is a very important piece to maintaining life. There are 4 basic mechanisms of movement across prokaryotic cell membranes. There's simple and facilitated diffusion. Those 2 don't require energy, so they're considered styles of passive transport. And then you have 2 different styles of active transport which do require an energy expenditure to move the item across. So simple and facilitated diffusion and active transport should be review. So you may want to review some of this material on your own. I will briefly describe it. So the concept with simple diffusion, that's just movement directly across the cell membrane. So just to remind you what diffusion in general is, diffusion is when molecules move from areas of high concentration to areas of low concentration, as you can see in these glasses. So here you drop a little bit of food coloring or a dye tablet into some water, and even if you don't mix it with a spoon or something or a stir stick, just by sitting and waiting, the random molecular movement of those water molecules is going to cause mixing. And random molecular movement is always going to cause things to spread out. So these dye molecules will move from where they're highly concentrated here to where they're least concentrated out on the edges of the cup, and that's going to keep happening over time until there's a fairly even distribution of dye everywhere in the cup. So as it turns out, random distributions are fairly even distributions. It's not perfectly even because it's random. It's not like you've individually placed each dye molecule equidistant from every other dye molecule. But when things spread out randomly, they're fairly evenly distributed. So you have random movement causes molecules to move from areas of high concentration to areas of low concentration until they're fairly evenly distributed everywhere. So you can see that happening here as well. So so at the beginning, you can predict where you find each individual dye molecule. If you took a little microscopic dart and you threw it into the glass, molecular sized dart, you would know where to toss it if you wanted to hit a dye molecule. You toss it right there where most of them are existing. But after about an hour, you wouldn't really know where you would find a dye molecule and where you wouldn't, because they're randomly and fairly evenly distributed through the glass. So that's diffusion, movement from high to low concentration. So simple diffusion is diffusion from high to low concentration across the cell membrane. So for example, the cell is always producing carbon dioxide as a waste product. So inside the cell, carbon dioxide levels are always high because the cell is always making more, making more, making more. Outside the cell, carbon dioxide levels are low because the carbon dioxide just floats away. So Carbon dioxide is nonpolar and it's small, so it can directly cross the cell membrane without the help of a protein channel. And it can cross the cell, and it's going to move from high concentration inside the cell to low concentration outside the cell. The cell doesn't have to expend any energy because it's moving down its the molecule, carbon dioxide, is moving down its concentration gradient, and that's the direction that the cell prefers it to move to get rid of it. Oxygen moves in the opposite direction. It's also a small nonpolar molecule. It can directly cross the cell membrane, the hydrophobic fatty acid tails of the cell membrane. So it doesn't need a protein channel to move through, but it's highly concentrated outside the cell and of low concentration inside the cell. And that's because the cell is always using up oxygen. It uses oxygen for respiration. So every time oxygen goes into the cell, it gets transformed to something else. It's no longer there. So oxygen concentrations outside the cell are always high relative to the oxygen concentrations inside the cell. So oxygen is always going to diffuse into the cell by simple diffusion. So simple diffusion means no energy is required because the molecule is moving from high to low concentration, which is the direction molecules always move, to increase randomness. And they don't need a protein channel to help them move across the membrane. Facilitated diffusion is high to low concentration again, so the natural direction of molecular movement from high to low concentration, so no energy is required. It's passive transport, but a protein channel is required. So that's going to be for charged or polar molecules. So salts have positive and negative charges. They're attracted to water. The water's going to pull them back from the hydrophobic cell membrane. They're not going to be able to cross without a protein channel that's hydrophilic that allows the salt to pass through. Same thing with sugars. So any molecule that has an electromagnetic charge of any kind is not going to be able to cross directly through the phospholipid bilayer. It's going to need a protein channel that will help it move across. But if it's moving from high to low concentration, it doesn't need any energy expenditure. Okay. So that's going to be things like acids. So a lot of metabolic waste products are acidic, and acids have a lot of hydrogen ions, meaning H+. It's got a positive electromagnetic charge. The cell wants to get rid of them. So they're building up inside the cell, so they're high concentration in the cell, low concentration out of the cell. So those acidic waste products are naturally going to move from high to low concentration from the inside to the outside of the cell. That's just what the cell wants. Doesn't need to invest any energy in moving it the direction it wants it to go, but it needs a protein channel because it can't move directly across the phospholipid bilayer. Okay. Now, molecules naturally move from high to low concentration because of this little concept of thermodynamics called entropy. So one of the laws, there's two laws of thermodynamics, and one of them is that entropy in a system is always increasing. Entropy is a measure of chaos or randomness. Okay? It's a measure of disorder in the system. So what that really means is that disorder is always increasing. So that's why things spread out randomly. So you start out with things being orderly. Here's where you find the dye, and here's where you don't find it. And then over time, it all spreads out randomly. You can sort of think of that as like the condition of your room. In your room, you start out at the beginning of the week with your pens in the pen jar and the socks in the socks drawer and books on the bookshelf. And then just by living in your room throughout the week, the books get distributed more evenly throughout the room. Your homework is evenly distributed throughout the room. Your socks are all over the floor, evenly distributed throughout the room. That's an increase in entropy, an increase in disorder, right? And that just seems to happen naturally without you even trying. What you have to do to reverse it is invest energy. You say, oh, I'm going to take like 2 hours and I'm going to invest a lot of time and energy to reverse this process of entropy and put everything back, compartmentalize everything so that the socks are all in one spot and the pens are all in the other spot and the homework is all in the other spot and the books are all in the other spot, right? So imagine what the cell would have to do. The same thing for the cell. If the cell wants to reverse entropy, it needs to invest energy. And that's where active transport comes in. So active transport is when you take molecules, that's when you have all the dye is already spread out in the glass and you want to reconcentrate it down into a tiny little dye pellet or dye drop, right? That seems impossible, right? But there's situations where the cell has to do that. So for example, you might have very little sugar outside the cell and a lot of sugar inside the cell, but the cell still wants to bring in more sugar against the concentration gradient. If sugar were left to its own devices, it would move out of the cell down its concentration gradient until it was even on both sides of the cell membrane. But the cell doesn't want that. The cell wants to hoard as much sugar as it can because that's its energy source. So even though there might be almost no sugar outside and a lot of sugar inside, it still wants to bring sugar in. That's reversing the process of entropy. It's reconcentrating things, and that involves an energy expenditure. So active transport, regular active transport, is where there's a protein channel, and the protein channel pretty much just grabs the molecule that needs to be moved against its concentration gradient and uses energy to bring the molecule in. And energy can be in many forms, but usually it's in the form of ATP, which we will cover when we get to metabolism. So ATP, just, you know, it's the energy currency of the cell. That's what we always say. But that's sort of like the payment, the energy payment. But it can come in any form. But the cell has to use up energy or pay in energy to move something against its concentration gradient, whether it's moving it in or moving it out. Group translocation is a special kind of active transport. Okay. Group translocation is a type of active transport in which a molecule is moved against its concentration gradient, and it's also changed chemically. In the process of movement, so that it's no longer able to fit into the transport channel, and that prevents its escape in the wrong direction. This type of transport across the plasma membrane is unique to prokaryotes. So all the other three methods of transport that we just covered, we'll go back here, simple diffusion, facilitated diffusion, active transport, that's both prokaryotic cells and eukaryotic cells can do those. Only prokaryotes can perform translocation though. So if you look at this, the cell wants to bring in sugar, okay? So sugar can fit perfectly in the receiving area, the docking area of this transport protein. And once it fits in there, the protein uses energy, it's not shown here, but it's going to use energy, use ATP, to move this sugar into the cell against its concentration gradient. But the problem is, if the sugar can fit into this channel, it could fit in the opposite direction too. If it can fit to go in, then it could fit to flow back out. And it would flow back out because everything moves down its concentration gradient from high concentration in the cell to low concentration outside the cell. So the cell doesn't want it to move backwards. So what it does is, this protein is actually a bit of an enzyme. It conducts a chemical reaction with the sugar on its way in and changes it to something that's close to sugar but not quite. So this is glucose 6-phosphate. Now, glucose 6-phosphate is a little bit chemically different from glucose, so it actually no longer fits into this transport protein. So now it's stuck. You know, if it was just glucose, it could have fit in this transport protein to flow back out. But now it's not actually sugar, it's not glucose anymore, so it can't fit back out. It still contains all those calories, but it's not the right shape to fit backwards through that channel. So that's group translocation. Now, so we've talked about the form, chemistry, function of the plasma membrane. And the integrity of the plasma membrane is essential to cell survival, which means if you want to kill a pathogen, you'll be sure to kill it if you destroy the plasma membrane. Bleach works just that way. Bleach works by disrupting the plasma membrane. And so does the antibiotic colistin. But like bleach, this antibiotic will kill any kind of cell, including a human cell, because all cells have a cell membrane. Every single cell, every single living thing has a cell membrane. So that's why you don't just drink bleach, right, when you have an infection. It's great at killing microbes. It's really, really good at it, but it's also going to destroy your esophagus and your stomach because it's going to destroy all the cells that make up those organs, right? So colistin is an antibiotic that people take as a last resort when they are infected by a drug-resistant bacteria. Good antibiotics should target anatomical features that are unique to the pathogenic organism so that they do not damage the host's cells as well. And we'll talk about that concept throughout the course, but when you're looking for a drug that kills a microbe that a human has to ingest, of course you know that you want it to be safe for the human but poison to the bacterial infection. Well, what you have to think about then when you're developing this drug is, what anatomical feature am I going to target? What anatomical features are unique to the pathogen so that I can target that anatomical feature without harming the host?